Good morning, everyone. Welcome to our Friday seminars online for a change. It is my pleasure to introduce to you our speaker today, Dr. Eric Locke from the University of Minnesota, who unfortunately couldn't join us in person today due to some logistical issues, but thankfully he's here online. So Eric is an associate professor at the Division of Biostatistics at University of Minnesota. He received his PhD in statistics from UNC Chapel Hill, and then also did a postdoc at Duke before joining UMM. And his research broadly focuses on data integration problems and their applications in systems and molecular biology. And he has done lots of interesting work in variety of data integration problems, which are of course very important and modern problems. And data integration problems could be from data, like integrating data from multiple sources, as well as data from, measured in multiple dimensions or multiple ways. And these typically have lots of strong applications in biomedical studies, especially omics kind of data sets. And Eric has a pretty strong research profile and very well-funded research profile, including lots and lots of publications, as well as lots of NIH-funded grants. And today he's going to tell us about some of his recent interesting work on data integration methods for so-called bidimensionally linked data sets and their applications in panomics and pan-cancer studies. So please join me in welcoming Eric. Eric, please go ahead and take over whenever you're ready.
Okay, thanks, Abhishek, and thanks to Raj for the invitation. I wish I could be there in person. I had planned to be as of yesterday, but had a flight that was delayed and then eventually canceled. And there's still snow on the ground here in Minnesota. So I really wish I could be there in Texas, but happy to do this virtually instead. And before I begin, I want to make sure I acknowledge my collaborators on most of this work are Junyoung Park, who is now a faculty in the Department of Statistics at Toronto, and Katie Hoadley, who's at the University of North Carolina, and helped a lot on the the cancer aspects of the applications I'll describe. So the title of this talk is Bidimensional Linked Matrix Decomposition for Panomics Pan-Cancer Analysis. If what I mean by bidimensionally linked matrices and by Panomics Pan-Cancer Analysis isn't clear to you now, I hope it will be at the end of my talk, but I wanted to start with just a simpler setting. So let's say we have a gene expression data matrix. What we see here is a heat map view of this matrix. In this case, the columns correspond to breast cancer tumor samples. And throughout this talk, we'll organize our sample set as columns and features as rows. So here the rows correspond to genes, and red corresponds to higher values, blue corresponds to relatively lower values, and what this is showing is the level of expression for each gene. So roughly speaking, just how active is that gene at this point in time. And the data here are high dimensional. We often measure at least several thousand genes. And when presented with a data set like this, a common first step is just to do a low rank factorization of the data. So when we do that, we're essentially factorizing this matrix into latent patterns, which are given by the sample scores and the sample dimension, which are then weighted by these gene loadings here. And with a rank R approximation, we can represent this as R column vectors cross multiplied with R row vectors. And there are several ways to do this kind of a low rank factorization in terms of principal components analysis, factor analysis, independent component analysis, and several other methods. Many of them rely on the singular value decomposition, which I assume we're familiar with. It's here that U corresponds to the left singular vectors of our matrix, V the right singular vectors, and D is just a diagonal matrix with the singular values on the diagonal. And one approach to approximating my data with a lowering factorization is to use a so-called nuclear norm penalty. So here we consider an objective where the first part is just minimizing the sum of squared residuals. So this is the Frobenius norm here. It's just the sum of squared entries. And the second part here is the nuclear norm, which is given by the sum of the singular values of a matrix. So this is the nuclear norm in general is defined as the sum of the singular values. And when we solve x hat here to minimize this objective, the resulting solution will have the same left and right data, except this is kind of a soft thresholding operator on the singular values, in that those singular values that are above lambda will be we'll subtract lambda, and those that are below lambda will just be shrunk to identically 0. So this does, if lambda is large enough, result in a low rank approximation where the rank is just the number of non-zero singular values after this operation. It also has some nice convexity properties over just the hard thresholding on the singular values, which is nice. One remaining question is how to pick lambda in an approximation like this. And one approach is to kind of motivate a choice of lambda by random matrix theory. So if we consider a context in which our observed data X are represented as a sum of low rank signal given here by A and independent normally distributed noise here given by the error matrix E, which we'll assume as normal 0, 1 entries, then the SVD for X, as long as the signal in A is relatively high, will most likely, those first R components of the singular value decomposition will likely be driven by the signal. And so that responds to these first R left singular vectors and right singular vectors. And our singular values will also primarily likely be due to the signal, but there may be some noise also that's just kind of along for the ride. And then after we've kind of covered the signal, the remaining singular values will solely be due to the noise. And there are results from random matrix theory that tell us that for a matrix like this, the largest singular value for the noise matrix will be approximately the square root of m plus the square root of n, where m and n are the dimensions of our matrix. So then one reasonable choice for lambda is just if we have standardized the data to have noise variance approximately 1. And there are ways of doing this that I won't describe in detail. But if we standardize things such that we assume that error has variance approximately 1, then we can just choose this upper bound as our tuning parameter. And what that will do, again, based on this kind of thresholding algorithm, is any singular values that may be due to noise alone, it will shrink those to 0. It'll also shrink the singular values in the signal to some extent, and it's conservative in the sense that this square root of n plus square root of n is kind of an upper bound on these error contributions. So this is a one theoretically motivated way to automatically select this tuning parameter. Now we'll use this to try an approximation of that gene expression matrix. So when we apply this nuclear norm approximation, the resulting rank is 18. So there are kind of 18 underlying components. These are the non-zero singular values that are contributing to this approximation. And you'll notice that, of course, the low rank approximation doesn't capture all the variation in my original data, but it does tend to capture a lot of the underlying structure, at least that's visually apparent in this heat map. So one approach to an approximation like this is just as like a denoising procedure, where we're trying to get at the underlying signal in our matrix. It can also be used, for example, for missing data imputation, where here, let's say, here I'm just arbitrarily randomly selecting, say, 30% of the entries to be held out as missing, but missing data are often encountered in gene expression platforms like this. So let's say we do have missing data here shown by the black entries in the heat map. Then one approach is just to solve a similar objective to what we had discussed earlier, except the least squares penalty is only on the observed data. So we're kind of putting a nuclear norm penalty on our approximation, but only considering the residuals for the data we observed. And when we solve something like this, we'll get a low rank approximation. Here's our resulting approximation, which then if we want to go actually impute those missing data, we can just find the corresponding entry in a low rank approximation and plug it in, which I'm doing here, which gives us a matrix that's not exactly the same, but is close to the original data before I artificially held out those missing values. So there are a lot of uses to a low rank approximation like this. Another is just as kind of a dimension reduction tool, in that with this approximation, again, I'm taking the M here maybe in the thousands, and I'm kind of reducing variability in my sample set to this matrix of sample scores, which is of dimension R, the underlying rank of my approximation. And in this case, we can look at, say, those first two components. Here, whether I use the nuclear norm approximation or not, this will just give me essentially the first two principal components of my matrix. Because recall, with that nuclear norm approximation, it changes the scale, but it doesn't change those right singular vectors, which are analogous to or equivalent to the principal components under kind of assuming our matrix is centered. So here we're looking at just those first two components of those first two sample score vectors here. And we notice that they're driven primarily by these colors, which represent clinical subtype in breast cancer. We have the basal subtype, HER2, luminal A, and luminal B. And I'll kind of foreshadow that this is the first of 4 times that we'll see these subtypes appear throughout the remainder of this talk. And so now what we've learned is that these subtypes are one of the dominant and latent patterns driving variability in my gene expression data. Now let's expand things somewhat, and let's assume I have what I'll call vertically linked data, where here we have a gene expression matrix, just like before. This is what I'll call X1, and then another matrix of microRNA data that I'll call X2. So genes and microRNAs are different but biologically interrelated things that are typically captured with different technical platforms. Just like there are a lot of genes, there are a lot of microRNAs. So both of these matrices will be high dimensional, and again, our sample set corresponds to columns. So here in the samples are matched here. So this is why we call it vertically linked. And it will essentially have for each sample a high dimensional vector of gene expression measurements and a high dimensional vector of microRNA measurements. And when presented with a kind of linked data set like this, if we want to do some kind of low rank approximation or dimension reduction like we just did, there are a couple of ad hoc approaches. One is to just do a separate factorization of each data set. But we know, like I said, there are biological relationships between these two data sets, which we won't be capturing in a factorization like this. We'll also, and for that reason, we may also have kind of a lot of redundant information in the scores here and in the scores here. Alternatively, we could just kind of stack these two data sets together into one large concatenated data set and do our factorization. Then errors that are shared across our two data sets. But that's not really accounting for the fact that these really are biologically two different things and may have their own patterns of variability. And they're also measured with two different technologies. And those technologies may have their own kind of technical variability that's specific to one data set or the other. So here, one approach is to model both of these at once. So here we model both what I'll call a joint component and individual components for each matrix separately. And here I'll use the terms joint and individual structure a lot throughout the remainder of this talk. And when I say joint structure, what I mean are actually variation that's driven by the same lower dimensional patterns in the sample set that are shared here across both X1 and X2. When I say individual structure, I mean lower dimensional patterns or a lower dimensional representation of the sample variability that explains a lot of variation in, for example, gene expression here, but not microRNAs. And here vice versa for the microRNA individual structure. And we find that by accounting for both these sources of variation, joint and individual, we're better able to capture what really is joint and what really is individual. So there are a lot of methods that do this kind of thing. I use the term joint and individual based on an article that discusses this method, JIVE, which stands for Joint and Individual Variation Explained. But there are a lot of other methods, including nice work by Dr. Gadanova that that capture this kind of a signal. And I won't discuss any of these methods in detail here, but we'll try a different approach. What we'll try is just extending that same nuclear norm penalty that I had for a single matrix and think about how we may extend that to this context. Well, kind of formally here, we've got our two data sets, X1 and X2. We'll try to model them with a joint part given by J here and an individual part given by A. And our goal will be to have kind of three low rank components. We'll have J, which is low rank, that's shared by the two data sets, so that'll correspond to joint structure. And A1 and A2 will be our separate low rank approximations to the individual structure. And here I'll just take that, that least squares objective and consider both joint and individual, and then put nuclear norm penalties on the joint and individual terms in my decomposition. And then by default, I can choose my penalty parameters for each term by the same kind of random matrix theory motivation that I use for a single data set. So that means for my joint part, because I have, in that case, if I kind of concatenate my matrix, I've got n samples and dimension m1 plus m2. That's the dimension of x1 and x2 here, which may not be the same. That's the dimension of my joint part. So I'll put a penalty that corresponds to that. And then for my individual terms, I'll just put a penalty that corresponds to the dimensions of each individual matrix. And then I can just kind of iteratively update by that same nuclear norm thresholding algorithm in my J, A1, and A2 until convergence. So this is a method that, again, there are a lot of approaches to do this. So our motivation at first wasn't to come up with yet another approach to this kind of problem, but we've actually been surprised at how well a simple approach like this works. And just to go back to our real data here, we have our gene expression matrix, our microRNA matrix, and we have the terms of our decomposition. Here's our joint structure, our individual structures, and our residual noise. Here, the rows and columns of all matrices are matched. So, this row and column here matches in each of these four matrices. And the columns also match again between the gene expression and microRNA matrix. This is ordered to show the joint structure. And so meaning the rows and columns are ordered by a hierarchical clustering algorithm to show the joint structure. And if we look at the joint structure, we do see that there are kind of patterns that are shared in both the genes and the microRNAs. For example, we kind of have this cluster of samples here that stands out, at least to some degree, in both data sets. You see that in the original data, but it comes into more focus with the joint structure. At this point, it doesn't look like there's a whole lot going on in the individual structures, but that's mostly just an artifact of how I arrange the rows and columns. So for example, if I were to take that individual microRNA structure matrix and reorder the rows and columns to based on a hierarchical clustering of that matrix, then things start to look a bit more interesting. I mean, and I see more structure. But if I do the same to the noise here, then it still looks like noise. So this really is just kind of independent, uncorrelated noise after I account for joint and individual structure. So that was kind of the heat map view. And each of those terms, again, is low rank, meaning there's a small number of latent components that's driving variation. So now what we'll do is say, look at those joint component scores, which are kind of those sample patterns that drive variability shared across genes and microRNAs. And when I look at those, I again find my subtypes. So this is the second of 4 times that these subtypes have come up. And what we've learned now is that these clinical subtypes have variation in gene expression, but that's also shared with the microRNA data. So this may potentially correspond to interactions between genes and microRNAs. That's a causal claim we can't make. But what this does tell us is that, anyway, these subtype-driven patterns are shared in genes and microRNAs, and we don't see these subtypes appear when we look at the individual structure for either data set. So whatever that individual structure is, it's driven by other factors.
Eric, can I ask you a quick?
Yeah.
Can I ask you a quick question? When you introduced the optimization method there, when the X1 and X2, the 2 parts, is there any way you're accounting for the fact that these 2 parts may have fundamentally come up from 2 different distributions? They could have been measured on very different kinds of individuals. So there could be some heterogeneity in there.
So that's a good question. And here there's kind of an assumption we're making, especially with the choices of these penalty parameters, is that for one, the noise level is similar in both data sets. And to do that, we actually have a prior step where we scale each data set to have, hopefully have similar levels of residual noise. But there's also an assumption here that things are quantitative, and this will work best if the noise is at least approximately normally distributed. And so that's kind of extensions of this kind of an approach that allow for other distributional forms. Say, one is normal, the other is Poisson or binomial, is another aspect which has actually been addressed by some of these methods, including this GI-PCA method in other work by Dr. Gaidanova. But here we're kind of, those are two assumptions we're making, is that things are approximately normally distributed, at least in terms of the noise in both data sets, and that the noise level is similar.
Right. I was just thinking more like if the genes, the X1s are your genes data, right? And your X2s are the microRNA data. Mm-hmm. So if they're measured onto very different groups of individuals, then they fundamentally will represent something different.
Yeah. So here, that is one assumption we're making, is that, again, the columns are matched here. So here, the samples, the columns are matched, meaning we have the same sample set for both data sets.
I see.
So that is an assumption we're making. I mean, this is also the utility of this kind of approach for missing data is also interesting if we do have some samples that are matched, some that are only for gene expression, some that are only for microRNA. And that's something we've done as well and explored, is kind of using the joint structure then to impute, for example, entire columns in the gene expression in microRNA. But in order to apply the method like this, you need to assume that at least one dimension is shared. Here, it's the columns that are shared. There's not necessarily a one-to-one correspondence between the microRNAs. Right. Does that answer your question?
Yeah, thank you.
It's actually a nice segue to what I was moving toward, which is, in this case, columns were linked. If we consider a horizontal linked case, it's instead the rows that are linked. So let's say we have a data set where, which we do, where we have tumor samples and normal breast tissue samples. And in a context like this, we have gene expression measured for both data sets for the same genes. So here it's the genes that are the common dimension between the two data sets. And we can think of doing a similar kind of decomposition, where we can find kind of joint structure along that shared dimension and individual structure to gene expression data, and the tumor samples and to the normal samples. And this kind of an application is also interesting, but here we're arguably most interested in say, the individual structure. Because what this then captures are patterns in the genes which explain a lot of variability in the tumor samples, but not in the normal samples. So in that case, we're kind of getting at hopefully cancer-specific variability in gene expression. We're not identifying factors that are just inherent variability across breast tissues. We're really getting at the cancer-specific variability. And then with the joint structure, we are getting at patterns in the genes which explain variability, say, across both tumor samples and normal samples. So before applying something like this, we tend to row center our sample sets separately. So we're not trying to get at, say, mean differences in expression between tumor samples and normal samples. That's certainly worth looking at, but there are a lot of other methods that do that. What we're trying to identify is kind of shared and specific patterns of variability to tumor samples and normal samples. So that's a hierarchically linked example. Now, finally, I'll get to what I mean by bidimensionally linked data. Let's say I have, again, gene expression and microRNA data for both tumor samples and normal samples. So then I call this bidimensionally linked because I have kind of some matrices that share row sets and some that share column sets. And if I think of doing the same kind of a low rank factorization or dimension reduction, there are actually a number of ways in which things can be shared here. So I've run out of space to fit this all on a single slide, but we can think about approximating this with fully joint structure. So here I've consistently using purple to denote things that are shared. And here when we say fully joint structure, we mean patterns that are associated across the genes in the microRNAs and explain variability across both tumor samples and normal samples. We have, say, row shared structure, or here, column shared structure, rather, in that we are identifying patterns in the genes here unrelated to the microRNAs, which explain variability across tumor samples and normal samples. And similarly here for the microRNAs. Then we have row shared structure, which the things are shared then this way and this way, which means we have underlying patterns in the samples which are shared by both genes and microRNAs, but not in the normal samples. So here we're kind of finding tumor specific gene and microRNA driven variability in here, gene and microRNA coordinated activity that's specific to the normal samples. And then we can have approximations that are individual to each of my 4 data sets. So this would correspond to variation here that's not gene variation that's not related to the microRNAs and is only in the tumor samples. And similarly for these other 4, for these other 3 approximations. And so that's kind of one view of this decomposition. Here I'm showing all terms. So we kind of have a global term, row shared term, column shared terms, and individual terms, and then noise, where each of these kind of 1, 2, 3, 4, 5, 6, 7, 8, 9 matrices will be of low rank to give that factorization. And to fit a model like this, this kind of a challenge was when we first started using this kind of a structured nuclear norm penalty. But first, just to define our more general framework, let's say we have matrices that are kind of organized on a grid. So again, this is, here we have it for a 2x2 example. Here we're generalizing it to account for kind of any number of row sets and any number of column sets. And here and going forward, the index 0 will be used to denote concatenations across rows or columns. So for example, here xi0 corresponds to the ith row set concatenated across all the column sets. And so in a framework like this, we can think of extending that same kind of structured nuclear norm penalty to this context, where I just fit the whole thing by a whole least squares problem where I minimize the sum of squared residuals, but then put a separate nuclear norm penalty on each of those low rank components. And then I can fix these penalty parameters just as I did previously, by just considering the dimensions that that component covers and fixing the penalty to zero out any singular values that may be due to noise alone based on those dimensions. And then I can just like it did previously, iteratively update each of these terms until the objective converges. And so I apply that to this data set, and when I do, again, I have these kind of 9 different terms that I can look at. But when I look for those subtype effects, I find that they're really only in the tumor-specific gene microRNA shared variation, which kind of makes sense. So here is now the third of 4 times that we've encountered these tumor subtypes. And what I've learned now is that these subtypes are driven by kind of shared variation across the genes in the microRNAs, and the patterns that drive distinctions between these subtypes are really specific to the to cancer tumors. They're not shared in normal tissue, right? So it really is kind of cancer-specific variation that that is causing these subtypes. Okay, so now let's expand kind of our view even further, and finally get to this idea of panomics pan-cancer integration. So like previously, I could consider breast cancer tumor samples for here mRNA expression is the same gene expression. So I can have a microRNA expression like you've already seen. There are a number of other so-called omics technologies. Like there's, I can measure DNA methylation for each tumor across many different methylation probes and so on. So I kind of have these different omics technologies, which are vertically linked this way. And then, whereas previously I just looked at breast cancer in normal tissue, here I can also consider multiple different types of cancer. So in addition to breast cancer, I can look at lung cancer, certain type of brain cancer, and so on. And so our goal will now be to apply a similar kind of factorization to this kind of a bidimensionally linked data set. And now there are even more ways in which variation can be shared. And I mean, for example, we could have variation that's shared between microRNA expression and gene expression, like we saw earlier. But those same patterns in the genes and microRNAs may also explain substantial variability in lung cancer tissues and in brain cancer tissues and in others, right? Or they may not. We may find a pattern that exists on genes and microRNAs, which explains variability across breast and lung cancer tissues, but not brain cancer samples. So really any possible kind of submatrix of this grid could have some shared pattern of low rank variability. And we're interested in identifying both those shared and individual patterns because it's well known that these cancer types have a lot of kind of molecular heterogeneity, which often corresponds to clinical heterogeneity and has other important factors. So the question here is, how much of that molecular heterogeneity within cancer types is shared across cancers from different tissues of origin? And so in we'll look at some data from the Cancer Genome Atlas, where we have here 6,793 total tumor samples that represent 29 different cancer types. So here, for example, BRCA corresponds to breast cancer, and so on. But these are 29 different cancer types, primarily defined by their tissue of origin. And we'll have data from 4 different omics platforms. That's gene expression, microRNA, DNA methylation, and protein abundance. And again, when we think about the possible ways in which low rank structure can be shared, well, it's really any subset of these 29 cancer types in any subset of these 4 omics platforms. So for example, we could have shared mRNA, which is gene expression, and microRNA variation across breast cancer, ovarian, and uterine cancer, or and so on. But when we tally that all up, it's in the trillions of possible combinations. So it's hard now. I can think about extending my approach similarly, but it's hard to simultaneously model all kind of trillion or possible shared structures. So let's consider an approach where that'll be our goal, but we'll try to be somewhat pragmatic about it, where we kind of have some upper bound, given by kappa on the number of shared, we'll call these structural modules. And each module is kind of a block of low rank structure that's shared across some of the row sets and some of the column sets. So here, the R and C here correspond to which row sets, here the omics platforms, and which column sets, here the different cancer types that module K is defined on. And so we'll kind of decompose variability in our full data matrix into a sum of low rank blocks that are shared across submatrices of my full data set. And to do this, again, I just put a nuclear norm penalty on each module that's, as we've done previously, proportional to, that's given by the dimensions of structure that module covers. So these are kind of the sum of the omics features that it covers and the total number of samples that module covers. And again, there's a lot of modules that we can estimate here, but ideally only some of them will be non-zero, right? So this may be sparse, and yes, we have a huge number of possible modules, but this may be sparse in that kind of with this kind of penalization, only a small number will be ultimately non-zero. And here I won't, for lack of time, go into our estimation method in detail. But what we essentially do is upper bound the total number of modules that we're looking for, and then have an algorithm that kind of seeks to simultaneously solve this objective while iteratively adjusting the row and column sets for each module to kind of maximize this objective, right? So we're kind of maximizing this objective over the row sets and the column sets to kind of find the decomposition into modules that explains the most underlying variability with this penalized objective. And so just a couple of theoretical results I'll touch on. With these kind of matrix decomposition methods of this form, identifiability and uniqueness are often major issues, and often in these decomposition approaches where we're decomposing things into shared and individual structure, orthogonality constraints on the different terms in the decomposition are used to guarantee that the decomposition into various components is uniquely defined, because it's not uniquely defined otherwise. But we've been able to show that with this structured nuclear norm penalty, kind of the mode, or kind of the global optimizer of the objective is uniquely defined under at least some conditions. And here, at least one set of sufficient conditions are that the underlying row and column spaces defined by the modules are linearly independent, which is a less stringent condition than orthogonality. But if we just apply this decomposition and then post hoc look and check if the underlying terms are linearly independent, then we know that this decomposition into joint and individual structure is kind of the unique decomposition that solves our objective. So uniqueness is one issue, and one other interesting result that I'll touch on is the fact that this objective also identifies a posterior mode under a Bayesian model. So with apologies to Raj and many others in the audience, nothing about my talk has been Bayesian up until this point. But we are able to show that this kind of structured nuclear norm objective identifies the mode of a Bayesian model, actually a pretty straightforward Bayesian model. We just assume normally distributed error, and that the factor component matrices in our low rank terms have normal priors. It's not an intuitive result, at least for me, but it's possible to show that that this the posterior density resulting from a model like this is proportional to the structured nuclear norm objective that we're solving, which brings up possibilities in terms of using the structured nuclear norm algorithm to identify the mode, and then perhaps applying some kind of Gibbs sampling to assess uncertainty about the solution. But I'll move on from that and just briefly, in the time I have left, discuss the application of this approach to our Panomics pan-cancer data. So first, what I'm showing here, I kind of capped the number of modules at 50. And we try to find the 50 modules that together explain the most variability or lead to the minimum of my objective. And when I do that, it actually doesn't lead to perfect sparsity, in that the variance explained by the 49th and 50th module are still non-zero. But after a certain point, they become almost trivial. And so we're able to capture most of the structured variability here in just a small number of modules. And here I'm showing those first 15 modules ordered by variance explained. So keep in mind that each module corresponds to low rank structure that's shared across some set of cancer types in some omics sources. And here you see the one that explains the most variability is completely universal, in that it's shared across all cancer types and all omics sources. And I'll focus just on a couple of these that I find intuitive. First, we have this fourth module, which is shared by messenger RNA, that's gene expression and DNA methylation, and some of the cancer types, but not all of the cancer types. And if I look at what that module corresponds to, here I'm showing, and again, this is low rank, and I'm looking at the singular values of that module. I find it's close to just a rank 1 signal that it's finding. Here I'm showing the first principal component on that structural module, and it's really driven by sex differences. So here we were in this component separating males from females primarily. And when we look at the corresponding loadings for that component, they're also intuitive. So here the red and blue here correspond to the X and Y chromosome, which are the sex chromosomes. And we see much more activity on those two chromosomes. So in terms of gene expression here, there's really only 2 genes on the X chromosomes that have this signal. That's the TSIX and XIST genes. These are responsible for so-called X inactivation, which is inactivating one of those X chromosomes in females, which then causes this different methylation pattern. So here, this is nothing new, but it's, I think, a nice illustration of what we're trying to find, which are patterns that may be driven by multiple kind of molecular variation at multiple levels, which explain variability across multiple kinds of cancers. And here, when we look at those, which of those cancer types were included in that fourth module, or rather which were excluded, it's those that are primarily on one sex or the other. So for example, breast cancer or ovarian cancer, prostate cancer, they're not in this list because the difference between the sexes is not a dominant pattern of variability for those cancer types. Right, so I think that's a nice illustration. One other module I wanted to highlight was this one here, which is only shared on BRCA that's breast cancer, and is shared across all 4 omics platforms. Are there any guesses as to what would be a dominant driver of variability in this module? So there we've again found our tumor subtypes. So this is the fourth and final time that we'll find our tumor subtypes. And now what we've learned is that these subtypes are not only driven by variation in gene expression in microRNA, but also in DNA methylation and the proteome. And that it really is kind of a breast cancer specific phenomenon, in that these patterns drive variability in breast cancer, but there's really not, for example, an analog of the basal subtype here in other types of cancer, right? So it really is, breast cancer specific variability, and so at least that's what the method would indicate. But just to double check that, I can take these same loading vectors across genes, microRNAs, methylation, and protein, and I can project the other data for the other cancer types onto those loading vectors to get the scores essentially for the other cancer types on this component. And here I find that really the black dots here are all other cancer types, and we're really not explaining much variability at all in the other cancer types. It's really only explaining the variability or corresponding heterogeneity in the breast cancer samples. Okay, so I'll end there and hopefully have a few minutes for questions. Again, here are 2 references, both on the BDFAC method and this BDFAC Plus, which was the last application I showed, and there's code available on GitHub here.
Okay, thank you so much, Eric, for the very nice and beautiful talk. I learned a lot. I'm going to open this up to the audience now for any questions.
Can I ask questions? Sorry.
Yes, Irina, please go ahead.
Jump in right away. Sorry,
No,
Eric. I know I can ask you more, but the time is short.
Sure.
It's the identifiability question, of course.
Yeah.
So I'm just trying to, so maybe you can go to the BDFAC, the original one.
Sure.
And this is because of another work we did recently. So here, yeah, I think this will work. Actually, if you go to the picture one, I think that will be more clear. Yeah, this
like
one.
This?
Perfect.
Sure.
Yeah. So here, when you do the raw sharing, it's like the sharing of the raw spaces. And when you do the column sharing, it's the sharing of the column spaces. And I know that you can construct examples where once you do the shared raw spaces, the column spaces kind of get modified as well. So it may depend on the order that you do. You can't always decompose them separately. So I was wondering if kind of extra restrictions, extra penalties that you put, if they take care of that, or if you have experienced
and I think that's one important point, is that this decomposition is certainly not identifiable in general, in that it's, like you said, that there are ways in which we can kind of rearrange the components across these various terms and yield the exact same approximation. So looking across, kind of this decomposition is not identifiable, but what the uniqueness result tells us is that with these penalty terms, it'll converge to a unique unique optimizer. So that's one way, for example, of defining a unique decomposition is just to take it as the decomposition that optimizes the structured nuclear norm penalty. Whereas there may be other ways, yeah, to enforce, and that's just one. But so it's not saying there's one unique way to represent this. It's just saying the structured nuclear norm penalty under some conditions gives one potential way to uniquely define decomposition. And if I, for example, change those nuclear norm penalty parameters a little bit, then I'll get another uniquely defined decomposition.
I got it.
And what we found is that, so it's not uniquely defined. What we found is that if we kind of simulate underlying signal that's kind of independent according to this pattern, we can still identify it pretty well. But if the noise is 0, that doesn't guarantee that we'll be able to identify these different terms exactly if we simulate kind of independent lowering signals.
Yeah. No, I understand. Do I have time for another question, or should I give someone else? I can wait. I can ask Eric later.
I have a question actually. So I was just wondering, when you are integrating these two platforms, do you have a data that is scale-free? Like
a data that is scale-free in that, yes, so that's the point, is that these two data sets, for example, genes and microRNAs, may be measured on very different scales. Mm-hmm. And they often are. Again, it depends on exactly what platform we're using, but the two data sets may be on very different scales at first, but then ultimately what we end up doing is a lot of pre-processing of these data sets in terms of often taking a log transformation and other things. And then before applying this kind of a factorization, we actually have a separate standardization step where we actually either standardize both data sets to hopefully have the same residual variance, or if we're acting conservative, we can just scale all the rows to have mean 0, variance 1, for example, which would kind of put these two data sets on a similar relative scale.
Okay, thank you.
Can I ask one question?
Yeah, go ahead, sure.
This is Yang. Very nice talk. I just had a question about earlier slides when you talk about missing data. So for this sort of pan-cancer, panomic data, I guess some of the missing data are informative. For example, maybe when gene expression they're missing, then maybe it's too low to detect by the technology. So the way you formalize as the product, I guess, just taking the Frobenius norm of the observed one, kind of assume the missingness was random, I assume?
Yes, that is kind of an underlying assumption. And that would be an underlying assumption both when kind of in the initial case where we're just imputing entries, where here you need to make the assumption that these missing entries are missing at random, at least. If you don't, then results will likely be biased in some way. So we do need to assume missing at random here. But I think that assumption, in a context like this, it's less likely to be met than in a context where, like I described here, we may be missing entire rows or columns. So say in this context here, we may be kind of missing some rows completely for gene expression and some rows completely for microRNA. And that's a common scenario, which often does happen somewhat randomly, just because you only have so much funding and can only kind of sequence or kind of apply the gene expression technology to a subset of samples for here, and then a subset of samples for microRNAs, and then it can be kind of random which are left out.
Yeah, I agree. If you have an entire row missing, most likely it's some constraint, budget or maybe technology constraint. But like I said, it's those sort of missing, not necessarily the entire row. For example, one gene is missing in this particular sample, that's perhaps more more informative.
Right. Yes, I agree. Often it is. Often it can indicate, lower expression, for
Lower
example,
expression.
depending on the technology. And yeah.
Yeah.
Right. I mean, my first question, like in the middle of your talk, was sort of like kind of catering to this kind of question that Yang just asked. Like, what if some part of the data, the fact that it is missing is because it is informed by some of its observed part of the data, like which is essentially like missing at random kind of a situation, given the missing part is somehow, the fact that you are missing has something to do with your features, the observed part. So that may be something to look into, like imputing informatively in some sense, right? Rather than just treating it something completely at random, which is exactly what I was trying to say when I said, are the two data sets heterogeneous in some sense? That would be also some evidence that there is difference in distributions in the two parts. But that was perfectly fine what you said. I realized that they are linked through the column, but in the more general case, right, this could be, the missingness could be sort of informed by the observed part of the data.
Right. Yeah. And one of the other point is that if we have, for example, like I was describing, if we're missing an entire column, then we can impute that with the joint structure, but for the individual structure, we can't say anything.
Yeah.
Right.
Because what we find is that when we apply this, it actually kind of shrinks to 0 in the individual structure, and it's just the joint structure part. Because that, if we have microRNA data, we can use that to impute the genes or vice versa. But if we're only looking at gene-specific variation and we're missing all genes for a sample, that we're out of luck.
Yeah, can I also add a quick comment? For those, for example, in this example, gene and the microRNA, the roles are sort of matched as well, right? Because you know what are the targets of certain microRNA to which genes. So that you have more shared patterns. So perhaps, I don't know, make things more interpretable. Or maybe
that's a good point, in that this, in general, I think with this overall approach, we're not using or claiming causal relationships in the result. But those could be, for example, used to the fact that, like you said, there are often genes that are known targets of certain microRNAs that could be used to inform things.
Okay, are there any other questions for Eric at this point? Well, okay, if not, let's thank Eric again for the wonderful talk, and some of us will at least see you later in the day.